a topic of great interest to me. So I skim over some details for time and I overdo some other details, but this is just scratching the surface of the topic. So, but one of the things you might ask, why would I talk about taxonomy as a subject? Why not something really interesting like the history of linoleum or Canadian sidewalk law? No, but seriously, the subject of taxonomy, the nuts and bolts of it, it's surprisingly little known, even among professional biologists. I get asked questions concerning the how naming occurs, why names are what they are, and such like that. So it's something that's not. I don't think it's covered as much as it should be. It's actually really interesting, and not just to me. It's interesting to a lot of people, and it's really useful. When you just understanding a few basics about how it works, you can learn a lot about the what a name means, what your species of interest, what it's about. You can learn from the name itself. So hopefully, I will cover or demonstrate some of these. So I want to go through a few definitions. Again, this is going to attempt to be brief. If any of you ever sat in one of my classes when I was teaching, brevity is not my strong point, but I'll do my best. So I'm going to start phonetic taxonomies. So these are sometimes called folk taxonomies. These are groupings of animals or groupings of organisms, not internal.
You're good. I'm just moving this out of the way.
Based on characteristics that may or may not indicate an actual close relationship. These can be things like the presence of fur, the presence of scales, a particular habitat. So they may indicate a relationship, they may not. So some examples: fish. The term fish is an aquatic vertebrate, typically with fins and gills. But all fish are not equally related to each other, and fish may not represent an actual taxonomic category. Reptiles, another great example. Scaly, cold-blooded vertebrates that are mainly terrestrial. But again, all reptiles are not equally related to each other, and the relationship to other vertebrates may vary. So, phonetic taxonomies. So a great example of this from the wildlife world, ungulates. You may have heard the term ungulate hoofed mammals. These are large hoofed terrestrial herbivores for the most part. But the even-toed and odd-toed ungulates, like the horse group versus the cattle group, are not closely related to each other at all. They're actually fairly distant within the mammals. And non-hoofed groups such as whales are nested within the even-toed ungulates. So again, the term ungulate's a good descriptive term for an ecotype. It's a good term to describe in sort of common parlance, but it doesn't indicate any close relationship necessarily. So looking at again, to illustrate phonetic taxonomies, this is a 2020 paper on the Panungulata, showing the fact like elephants, for example, are nested in between the even and odd-toed ungulates. So now, looking at phylogenetic taxonomies, this is the majority. This is the accepted taxonomies that are used in modern times for those sort of phylogenetic taxonomies. These are cladistic taxonomies. This is grouping animals based on their relationship, their common ancestry. They may or may not have shared traits, but they're genetically, they're morphologically, possibly related to each other more than they are to any other group of organisms. So again, using back to my whales again, or back to the whales, you can see that within the artiodactyls, the even-toed hoofed mammals, Whales are closer to hippopotamuses and even closer to the ruminants like cows and all that than they are to pigs and camels. So, let's see here. So it's kind of important to know that these are not mutually exclusive necessarily. There's kind of a trend in, I guess, pop science to sort of contrast old versus new, or looking at in taxonomy, phonetic taxonomies, how they used to do it compared to how we do it. But one's not necessarily worse than the other in sense of that one's more accurate, but at the same time, phonetic taxonomies, for example, I'm a groundfish biologist. Groundfish is not a taxonomic term. It includes flatfish, it includes rockfish, it includes things that are not necessarily related to each other, but they sort of do some of the same things. So there's a purpose behind using the term groundfish. It's not some archaic term.
Got a question. Is phylogenetic taxonomy exclusively defined by genetic makeup, or is there anything else?
No, phylogenetics can include morphological taxonomies as well.
Okay, so
It's just relationship. It's
okay.
Yeah. Now that is determined almost exclusively via DNA in modern times. But phylogenetics just means that I'll get into some detail on it. But
Okay.
it gets into this. It's the relationship, sharing common ancestry. So again, some definitions. Clade, you'll see this term a lot in taxonomic literature. A clade is a group of organisms that are more closely related to each other than they are to other organisms. So in modern taxonomy, a clade is roughly synonymous with categories above species. So a family is a clade, and then expanding that out in order may also be a clade. So a clade has to be monophyletic, meaning they share a single common ancestor that is not shared with members outside of that clade. So you may have seen these. This is called a cladogram. This is where each node in the cladogram represents a divergence of a clade. So I'm going to fill this in. Just keeping it very, very simple here with some fish and reptiles, birds and mammals. So again, I'll talk about some of the history, but Pisces was defined as a taxonomic category in the 18th century. Linnaeus described Pisces, but you can see from here that Pisces, they do share a common ancestor, but they share a common ancestor that's also shared with things that are not fish. So by this definition, these are not a clade. Looking at reptiles, another Linnaean taxonomic category. You can see the lizard and the American crocodile, they share a common ancestor, but they share the same common ancestor with birds. That's not considered a reptile. So this is also not a clade. But then to contrast that, looking at what's called the amniota. So these are tetrapods that basically use costal breathing. They have an amniotic egg and costal breathing. These are a true clade because if you go down the nodes, their common ancestor that they share a common ancestor that's not shared by any outgroups or groups that doesn't belong to that clade. So brief history. So the first taxonomies in the ancient world were known as utilitarian taxonomies. This was simply functional, describing things based upon things you can eat, things you can't eat, things in the ocean, things on land, plants for medicine, plants for grains, again, animals suitable for sacrifice, animals that are not suitable for sacrifice. Some of these taxonomies, if you look like there's a lot of the world of Etsy, great reprints of some of these what are called bestiaries, these old taxonomic, is probably the wrong term for them, but these where they have very detailed drawings of real things and actually got species correct, mixed with mythological creatures and stuff like that. They're just amazing, some of these prints. But as we move towards the modern era, we got Gaspard Baldwin. He was a Swiss physician and botanist, pre-Linnaean. He again, like most people of his time, used utilitarian taxonomies. But he, as a botanist, he described plants for in terms of utilitarian. All spices were in. He put them in a group called aromata, regardless of the relationship. But what he started doing that's unique is he started grouping to some domesticated plants based on flower structure and comparing it to wild plants and saying these flower structures clearly show a relationship. So that was one of his big contributions, was looking at morphology. And he also started using two names. He used a genus name, where it wasn't called a genus name at the time. Again, with what would become a species name. So this binomial system that was used later on, he developed in part. Now we get to the big one. Everybody's probably heard of Carl Linnaeus. Pretty amazing guy. He was a Swedish physician, botanist, zoologist. Okay, maybe not that one. But he is the father of modern taxonomy. And for good reason. So he formalized the binomial classification system, that's genus and species. And he also, in some ways, maybe more importantly, developed a hierarchical system, family, order, class, phylum. Those were all Linnaean terms that he developed and defined. I want to, without going into too much detail, I just want to kind of better give you guys the some information on Carl Linnaeus to show how remarkable he was. He was the first to classify whales as mammals, which seems pretty obvious, but even more than that, he actually, by dissecting a porpoise, suggested that whales were closer related to ruminants based on their digestive system, something that was ignored for centuries until genetics showed that Linnaeus had it right. So that's pretty amazing. Almost prescient in some ways. He was the first to classify humans as primates. This was pretty heretical at the time. He developed an early bias-free morphometric analysis, meaning he took suites of characters and looked at them and analyzed relationships based on those. Very crude by modern standards, but pretty amazing when you look at some of this stuff. And he formalized anatomical naming systems. This means that for each different taxon, for example, crustacea, chelae, he like developing terms that were specific to a group and saying this is all the terms we will use, so it's standardized. So every natural history book's not using different terms for the same structures. He started formalizing those and defining them. Which kind of a little bit of an aside, I'm not going to go into too much detail, but some of his terms are still used to this day, like for crustacea. Others are not. He did an entire chapter on bivalve anatomical naming system that was declared obscene at the time for good reason. And it was quickly abandoned. There's a lot of articles on another mollusk biologist, Da Costa, viciously attacked him and tried to get the Pope involved to like have him shut down for his obscenities. Pretty interesting. Now, Charles Darwin. So this is where my brevity could go out the window. But we'll keep it. Charles Darwin was an English zoologist, naturalist, and geologist. His recognition of natural selection and universal common descent is the basis of modern taxonomy. Again, between Linnaeus and Darwin, there was a sort of a push to look at relatedness, but what does relatedness mean if you don't have a mechanism by which something can be descended from another organism or have a common ancestor? So Darwin provided that mechanism. And he drew arguably what's the first cladogram. This is actually on a scrap of paper in one of his notebooks that was lost. And then it was rediscovered by Tim Barra. Found it by buying old books. He collects old Darwin books. And he actually got a notebook, and this paper was finally rediscovered after almost a century of being lost. But it's the very first cladogram. And I like that he wrote, I think. I kind of like that. Okay, so now we get into the modern era, molecular systematics and cladistics. So it started actually in the 19th century. May not think because again, we associate this with genetics. But in the 19th century, they were using pigments, like plant pigments, and suggesting that certain algae, brown algae, have different pigment suites than red algae do, and using this chromatography, paper chromatography, as species, can look at species, look at different groups. In the 1950s to the 1970s, proteins and then additional micromolecule analysis, things like serology were developed for taxonomies. And then, of course, now it's almost all DNA. The advantage of using these molecular analyses, whatever kind you're using, is that they're potentially bias-free. If you're looking at two animals in your hand, you already kind of think, I think this one's more closer than this one related to each other. And so your observational bias. But if you're looking at tables of numerical sequences or stuff like that, you may be less biased. So, okay, just a quick aside on molecular systematics. It's an extremely valuable tool, and it's only getting better and more accessible. I mean, when I was in graduate school, DNA analysis was available, but phenomenally expensive and out of reach even for university. But just I think this is a fun example I ran across when I was putting this talk together. The first whole sequencing of the human genome was completed in 2003, and it took 13 years to do a single human genome, and it cost $300 million. Guinness Book of World Record has now the fastest sequencing of an entire human genome is just over 5 hours, and that particular one cost about $1,000. So it's amazing how the technology has advanced. So again, sequence data is essentially a blind study. But here's where it can, I guess, not be entirely relied on, is that it depends on that the sample was taken, that the species the sample was taken from was correctly identified. There's a database called GeneBank where these genetic sequences are housed, and they're available to other researchers worldwide. But a lot of those sequences are misidentified. So they were taken from something. Well, we encountered that. I worked on a project looking at krill. They were doing eDNA studies on whale scat and identifying krill. And so we had krill species, but one of the krill was identified as a pink shrimp. So one of our samples came back as a pink shrimp, and it clearly wasn't. So things like that can occur. And hopefully, as this becomes cheaper, more available, more sequences are added, those kinds of mistakes are being kind of swamped over just by numbers. Okay. So cladistics, this is the recognition of natural groupings. This means true clades that may not be easily ranked via a Linnaean system. 25 years ago, it was predicted that Linnaean categories, meaning the term family, order, class, would become completely obsolete, and we would only talk about clades. But this wasn't very practical, and we love our categories. So you'll still see now modern taxonomic literature still uses Linnaean ranks. And a balance has been accepted. So you can say this is a member of a particular family, but know that that word family doesn't necessarily specify the exact relationship of the individuals in that family. We know that it's artificial, but we can still use the term because it's convenient. So there is an organization called the International Commission of Zoological Nomenclature, the ICZN. I don't know how well known they are, but they are approximately 30 different countries. They have offices in 30 different countries. They're an amazingly powerful agency, surprisingly enough. They regulate the scientific naming of animals. These are the people who can declare a name invalid if you hear about, well, that name's no longer valid. The ICZN is the one who can make the final determination if a scientific name is valid or not. Just to point out, they operate in parallel with, but they're not connected to directly other nomenclature organizations. For example, the ICN-AFP, which is the International Commission on the Nomenclature of Algae, Fungus, and Plants. Or the ICNP, which is for prokaryotes, commonly bacteria. So different agencies, they have their own rules. The rules are very similar in a lot of cases, but they have they're not dependent on each other. So the ICZN is the final authority on this. They actually operate almost a court system where disputes on names can be taken to them. Like if two researchers name something at the same time, which name is valid? It's the same species, but they use two different names. The ICZN can go through and determine who published first or who technically had it first, that kind of thing. So they have some rules. There's a bunch of rules that they publish. It looks like our reg book. It's a gigantic volume of all of the rules, and they vote on different amendments and things like that. Pretty complicated, but some basic ones. Priority: the oldest available name is valid. So whomever comes up with the name first, that's the name the species will have. There are some exceptions to this, but for the most part, priority is pretty critical. So I'm going to give an example of this. The gooey duck clam. Those shellfish people, like we know, this was known as Panopea generosa for a really long time, described by Gould in 1850. But then in 2000, Cohn et al., looking at a fossil gooey duck from California, determined that the fossil gooey duck was the same species as the living gooey duck. But because it was published in 1849, it became the species for the gooey duck. So the species name went from Panopea generosa to Panopea abrupta for about 10 years. And then it was determined, further analysis showed that the fossil species was indeed distinct enough to qualify as a species. So it's gone back to generosa. What's it?
Just joking.
This can get really complicated. So the priority, binomial nomenclature. So species names are always binomial, genus and species. If there's a subspecies, it becomes a trinome, 3 names. All higher ranks, that's family, order, class, phylum, are uninomial. And then this is a really important one. This is the principle of homonymy. The names of each taxon must be unique at all levels. That means unique species, genus, everything has to be, you can't have a genus and species combination that's the same for two different organisms. And this is why a lot of times names, when people ask why do the names keep changing, a lot of this is the principle that's responsible for that. And again, another example. So this was named by Linnaeus, cancer strigosus. Now you may look at that, but familiar with cancer crabs, know that this is not a cancer crab by any stretch. But at the time when Linnaeus described, all crabs were in the genus Cancer, regardless of how we know them now. So he described this galathioid as a cancer strigosus in 1761. Then, either through ignorance or whatever, for whatever reason, Herbst described in 1799 cancer strigosus, a Mediterranean shore crab, different species altogether, but used the same name. This invalidated, Herbst's name became invalid because it couldn't, this principle of homonymy. So it became renamed by Latreille, renamed it Cancer alboliniatus. Then the genus was moved from Cancer to Galathea strigosa. So technically, this Cancer alboliniatus could have gone back to strigosus again. So it could have reverted. But as it turns out, Herbst, his type specimens, the actual preserved material was non-existent. So there was no way to prove that that was the species that is represented by the modern. So Latreille won out on that, and now it is known as Graspus alboliniatus. And these things can switch back and forth. If a genera changes, sometimes it will bring something into a homonymy that wasn't before because the genus changed. So now it's a homonym. So you have to go back to the next oldest name, these kinds of things. So it can get really complex. So designation of a type. So every described species, supposedly, has to have a type species, a type taxon. These are what these are is like if you are going to name a new species, you'll collect specimens and you will archive those in a museum somewhere and say this is the type. This individual is the one by which all others have to be compared to in order to be valid. Typically, it's not just a single type. There's a single type specimen, but then there's also what are called paratypes. So you'll have a range of them. You might have males, females, juveniles. You may have a whole range of species to define it. So again, these should be in permanent archived specimens. I mean, these should be permanent archived specimens. There's a big controversy going on right now about phototaxonomy, which is if I take a picture of it, and then I describe the species based on the photo, does that count? So there's a lot of, there's actually an ongoing debate as to whether or not phototaxonomy, if those names should be valid, if something's described from the photograph. So these are what all the others are compared to. So if you are describing a new species, you might request the types from museums and say, actually have them sent to you to compare to yours to see if they're the same thing or to see if they're closely related. You might want to look at the type specimen of the whole group of them, everything in the genus. You might want to look at those. So when I did a deep sea coral project, we were looking at some deep sea crabs, and we've got the type specimens from the LA County Museum, and then we're to verify that ours were the same species, not some new species, because the types were described from Southern California, and ours were from Alaska. So that's an example. So we looked at the types and went spine by spine and said, yes, these are the same species. So a few other fun rules. You can't name a species after yourself. So when a common question people when they hear about a new species described, they'll say, well, can you name it after yourself? And that's the end. You are not allowed to. There have been some, I don't know, controversies over researchers. There's a mollusk biologist named Oldroyd who named stuff after his brother. So, who was not a biologist,
Same last name.
same last name. While claiming that it's named after, like it was all named after his brother, but it sort of seems like he named a whole lot of species with this same last name. You can't sell the name, so you can't find something new and then auction off the right to name it. That's not allowed. The name should not appear in print. This is an important one. This has happened more than a few times where I find a new species of coral out there, and I think I'm going to name it, I don't know, Loxotholecus olsoni after Andrew, and I tell everybody about it, and then somebody types it up in the newspaper article. That name now becomes the official description, and the author in that newspaper article described the species because it's in print. So I'm no longer the author. That can be disputed, that can be taken to the ICCN, and they can have that name removed, but then I have to come up with a different name, or another researcher can scoop it and redescribe it. So there's things like that. That's why when somebody is describing a new species, they tend to keep the names to themselves. So the structure of biological classification. So again, I think most of us learned this in school. These ranks. But again, these are not necessarily, the real world may not fit into these categories as easily as we would like them to. So if you'll see this a lot if you look at classifications. You'll see these sub and mega categories. You'll see superfamily, subfamily, infraclass. These kinds of designations are an attempt to actually fit reality into this Linnaean taxonomy. But I want to again make it clear this is artificial. So here's just a quick example here. So within the Crustacea, so class Malacostraca, order Decapoda, suborder Pliocindata. There's actually more categories I did not include in this. And these are the Pendalin shrimps. These are commercially harvested shrimps in Alaska. This is 8 species. So all in the same genus, but 8 separate species. So I'm going to talk about how this is some of the importance of this, and I'm going to use my favorite group, which are the cowries. So I'm going to start with Cypria tigris, the tiger cowrie. I picked this one because you may not know it, but you probably, or somebody you know has one of these in their home. These are sold at every single tropical gift shop in the world. They're abundant and common. Yes, so Cypria tigris, described again by Linnaeus, 1758. So the first thing, the genus Cypria, at the time, it's not the case anymore, but at one point in time, all cowries were in the genus Cypria. So a single genus named by Linnaeus included all the cowries. Now, this is actually one of the few members of it that's still in the genus Cypria. Most of them now have been moved to other genera because, again, they look similar, but they're distantly related to each other. So the species Tigris, this separates it from the other species that Linnaeus described, which is Cypria pantherina. Somewhat similar, but different species. And again, the name Linnaeus, that's the author. That's the person who described it, and then the date the publication came out, which is 1758. But to complicate this a little bit more, here we go. There we go. So about 30 years after Linnaeus, Shaw described a subspecies, Cypria tigris pardalis. Now, it wasn't known at the time, but now we know that this Cypria tigris only occurs in the Indian Ocean. The same species or the same types of shell from the Pacific Ocean are all pardalis. So there's a break, meaning that they speciated when they entered the Pacific Ocean at some point in the distant past. So what does that do to the name? So now you can see this is a trinome, because there's genus, species, and now a subspecies, pardalis. So what this means is that Cypria tigris has to become Cypria tigris tigris. So when you see a trinome, you know that there's a subspecies. Even if it's duplicated, that means this is called the nominate subspecies. So it's Cypria tigris tigris as opposed to Cypria tigris pardalis. And now actually there's two others. There's Cypria tigris schilderiana and Cypria tigris lorenzi, but I'm not going to talk about those. So this is kind of a fun example of them. I use this one. This is just kind of showing off because this is one of the world's rarest cowries. It's in my collection, so it's just like showing off my Ferrari. So this is Nesiocypria terramaki, Kuroda, 1938. So I'm going to kind of break down the name here. So first of all, you see it's a trinome, so that tells us that there's a subspecies also in my collection. Nesiocypria terramaki polyphemus. But to get back to the naming convention here, so you'll see that name Kuroda. That's the author, that's the last name of the scientist who described it. And then it's in parentheses. So when you see the author and date in parentheses, that tells you that it changed genus, that the genus was not originally Nesiocypria. So whenever you see that in literature, you'll see it. Sometimes you'll see this if you do editing. People will put all authors' names in parentheses, like their citations, and that's not correct. If the genus hasn't changed, it should never be in parentheses. So this tells you that it was changed because it was originally, Kuroda described it as Cypria terramaki. So this is one of those examples of where all cowries don't belong to the genus Cypria. So again, keeping this here. So people names. You'll see that the species name ends in an I. That tells you a lot. That tells you that it was named after a person. And because it's an I, it tells you it was named after a man.
Now, a quick question on that.
Yeah.
I see that with the double I, and I hear some people say terramaki I. Is that just wrong, or is that sort of just out there?
I think it would depend on I'd say terramaki.
Yeah.
But yeah, that could be
Just like
correct.
hearing you both of the eyes, like Oncoricus clarkii or Oncoricus clarkii, I've heard. But
That's a good question. What I do in those cases, and unfortunately with a lot of these names, I never hear them said.
right,
I only read them. So like when it back up like Oncorhynchus, like I actually listened to a Korean researcher say the name Shashua. And I'm like, that's how you pronounce it.
Oh, yeah.
You know.
Freaking Xander.
So like after you see this Tawashi, whatever. That Shashua is how he described it. So I'm like, well, I'm sticking with that. He seems to know what he's talking about. But anyway, so this was named after Akabumi Taramaki, a shell collector who helped organize the Emperor Hirohito was a shell collector, and this is one of the people who organized his collection. And so you'll see a lot of species named after Akabumi.
And one other quick question, if I can, that you mentioned the rule about not naming it after yourself.
Yes,
Has that always been in place, or were there a few that snuck in there kind of before the IC?
I would not be surprised if that happened, but it's been, I mean, I think as a convention, I think that's been around for a long time. Before the ICZ,
Okay,
just because you would, otherwise you would think some of the more arrogant researchers would be able to
Yeah.
things named after them, and you don't see that.
Yeah.
Because the ICZ, I think, was founded in 1850. There's a lot of species from before then, but. So that's great question. I should have looked at stuff I should have looked up. So keeping with that, here's another cowrie. I see a lot of cowries. Palmula cypria katsue, also described by Kuroda in 1960. And again, you see it's in parentheses, so you know it changed names. This was named after, is because the AE was named after a woman, and the wife of Akabumi Taramaki, she was also a well-known shell collector. So naming after her. So you might look at it and go, why didn't they name it, since you're using like Taramaki or Taramaki-I? Why didn't they name it Taramake? But remember the homonymy, even though the gender is different, it would still be a homonym, the cypria. It could be done today because these are now in separate genera, but when it was described, he chose to go with her first name, so he went with Katsu. So we named it Katsue, Cypria katsue. Now it's Palmula cypria katsue. So again, keeping with people names, you'll see this aurum at the end of the scientific name, the species name. This tells you that it was named after a couple or a family, not just a single individual. So Angiorum was named after the Angio. So you'll see that aurum that tells you that it was named after more than one person. So a lot of times people will honor a family instead of just an individual. So place names. So this is actually a really fun gallery, independent of this talk altogether. This is actually the group it belongs to are the oldest known cowries. Then the first fossils of them are from the late Jurassic. So these are actually really ancient, and this is truly a living fossil, the Propastellaria. But you'll see the name is Surinamensis. You see ensis after a name, it tells you that it was named after a specific locality, often where it was first described from or where it's well known from. One thing to keep in mind with this is that because of how taxonomy and names shift, you can't always rely on it as you know that was named, why it was named after that, but it doesn't mean where you're necessarily going to find it. There's a cockle common in Alaska. It's Clanicardium californiensis. It's absolutely not found in California. Found in Alaska, and it's because at one point in time it was a single species found all the way down to Southern California. Then the Southern California ones were found to be not related or different species, so they were described as something else. So it left ours, because ours were the type lot, were from Alaska. The name Californiensis is stuck with it, even though they occurred to northern British Columbia up through the Gulf of Alaska. So you can't say just because it has the name on it where it's from. But in this case, Surinamensis is legit. Name at a time. Shoot, getting close. Holy smokes. Luckily, I'm almost done. So habitat names. So Bistolida clavicola. Again, it's a trinome. You know that there's multiple subspecies of this one. But that cola, that means home of or lives in. So clavicola tells you when you see cola, that's describing a particular habitat. In this case, clava refers to keys as in the geographical feature. So islands. So this is found on offshore islands off of off of East Africa. So clavicola, key living. Some names are descriptive, and this is pretty common. So you'll see like in these calories, Rancianella punctata. Punctata means spotted. And piriformis is pear-shaped. So, a few. Unfortunately,
I think I shrink that.
okay,
You just keep talking.
okay. Okay, sorry about that. I can see it on here. I just didn't realize it's blocking. So a few useful notes. This kind of just might help for writing, whatever you're doing. So genus names are always capitalized and species names are always begin lowercase. You'll see this done wrong extensively, especially in like things like newspapers or in pop science articles. So this large carnivorous dinosaur named Allosaurus atrox, yeah, that's incorrect. It should be lowercase, the species name. Skip one.
Oh, hold on.
What did I do here?
Okay, you're good.
Okay, there we go. All right. So when you see the abbreviation SP after a genus name, that means that you're looking for you're looking at a single unidentified species of a known genus. So an example, also present in the drift line were the remains of an unidentified Scyphomedusa chrysaroa sp. That means you didn't bother to identify it, or you didn't want, or didn't, whatever the case may be. For whatever reason, you just know that it was a chrysaroa. It may not have been the target of your study, so you're not looking at that. You're just pointing out that there was a chrysaroa, a single species present. When you see the abbreviation SPP, this means that there are multiple species of a genus. They were noted but not listed. Again, that may be outside the scope of your study. You're just noting that they were there. So, an example, the bycatch in this fishery is comprised mainly of soft-nosed skates, Bathuraja SPP, and Phycoplistus rockfishes, Sebastes SPP. So, you could have identified them, you didn't, because that's not the purpose. You're looking at a target species, so you're just pointing out that you know the genera that you found, but you're not listing the species. So I'm going to end this with some fun names. This is an extremely rare. It may be extinct. I don't think they've been found. The actual living specimens haven't been seen since the 1950s. That they were described from preserved material. Bah humbugi. So, more of these here. There's a fossil snake named Monty Pythonoides Europaeus.
That's legit. That's a real name.
Yeah, unfortunately, it was synonymized with the genus Moreira now. So, and Moreira takes precedence, so the name has been dropped, but it was described as Monty Pythonoides.
And the ICNZ went that through.
Yeah. Well, there's some really good ones, but so this is Polystemus chewbacca. There's also a Polystemus vaderi.
Oh my goodness.
More here. This is my favorite, absolute hands down my favorite. This is a fungus beetles that feed on slime molds. The genus is pronounced jelly.
Oh, that's cute.
Yes, because they feed on slime molds, jelly. It's legit. However, it's more fun because there's 5 species described by Miller and Wheeler. There's jelly bean, jelly belly, jelly donut.
No,
And these are legit. These are still extant, legitimate species.
we got not a party.
Jellyfish and jelly roll.
That's adorable.
Isn't it? And these are just like a millimeter long, they're very small beetles found in the neotropics, up into Arizona, actually, so if you make it to lower southern United States, you can collect your own jelly. Okay. This is Irwin 2000. Just as an aside, Irwin, the author of this, if you've ever seen movies or in popular science where people are fogging like the rainforest canopy and then collecting all the dead bugs, and that the estimate of 30 million species alive on Earth, Irwin's the one who came up with that. He was a beetle expert. But this particular one evaded capture. He had some damaged specimens and attempts to find intact specimens evaded for a long time. So when he did find some, he named it aggravation. But this guy was a lot of fun, as he also named in the same genus Agra Kate Winslet, Agra Schwarzeneggeri,
Yeah.
Agra Beeges, Agra Sasquatch, Agra Yoda, Agra Memnon, hilarious, and Agra Cadabra. So, and that is it. I'm gonna leave you with a quote from Linnaeus. And if anybody has any questions.
That's too funny.